everybody. Okay, today is really the midpoint of this course. We're ending our excursion in the infected cell. We're going to go next to how viruses cause disease and how we can prevent it. But today, the last vision of an infected cell, I want to give you an overview of what happens based on a lot of what we've talked about so far. So we've so far talked about gene expression, genome replication, assembly. We've all done this in infected cells. And as you know, those activities depend on a lot of host functions like metabolism, biosynthesis, signaling, and trafficking. And today I want to give you an integrated view of what happens with all of these systems in a virus-infected cell. And so I'm going to cover 4 different areas today. I'm going to talk about signal transduction pathways and their relationship to virus reproduction. I'm going to talk about gene expression, that is the production of mRNA and then protein synthesis. I'm going to talk about metabolism, and then we're going to end with a little discussion of how cellular organelles are remodeled in virus-infected cells. So start with signal transduction. As you know, cells need to respond to their environment. Whether they're in a dish in the laboratory or in an organism, they need to know what's around them. They need to know the nutritional environment. So these are governed by signaling pathways. There are typically receptors on the cell surface for, say, a growth factor. A cell will not divide unless growth factors are present. And so there are receptors typically on the cell surface for growth factors. And when a growth factor binds such a receptor, it initiates a signaling pathway. Now, what is a signaling pathway? It's a series of events from one protein to another, typically phosphorylation, that end up having an effect. And so here, for example, in this signaling pathway, we have a growth factor receptor at the cell surface. And then if the growth factor binds, the green line means a positive effect. So that would activate this PI3 kinase, which would then activate GTPases and so forth. And then in the end, for example, you could stimulate mRNA translation, you could stimulate cell growth as a consequence of a growth factor binding. And so in these pictures, I'm going to show you a green arrow means stimulate, but these pathways also have inhibitory parts, so the red arrows mean inhibition. Now, viruses tweak these pathways to their benefit so that they get more efficient reproduction. And you can see on this slide, a number of different viruses are shown to either enhance or inhibit particular aspects of the pathway. So we're going to look at that now to see how some virus infections interfere in signaling pathways to promote their own reproduction. But these, again, are normal parts of the cell. They're there for cell processes, and so virus reproduction simply taps into them. So here are 3 examples of how signaling via PI3 kinase, phosphoinositol kinase 3 kinase, it's a very active kinase in the cell that is involved in a lot of signaling pathways, typically downstream of some receptor interaction. So first on the left, we have adenovirus binding to a receptor on the cell surface, 2 receptors if you recall. It starts to be endocytosed at the very cell surface. And the interaction with the fiber receptor initiates a signaling pathway shown by these green arrows. And you don't need to know the details of the pathway, except RAC, of course. Racaniello protein. It's not really, but I like to see it that way. Eventually, what happens is this signaling pathway loosens up the actin microfilament layer just beneath the plasma membrane. So just beneath the plasma membrane of cells, there's this actin layer, and if you don't loosen it up, you can't do endocytosis. So this is a normal pathway that cells use to allow endosomes to go in, to loosen up that actin microfilament. So the virus is doing this by binding the receptor. It loosens up the microfilament, and therefore it can be endocytosed. So that's one example. In the middle, we have influenza virus binding to sialic acid receptors. It's doing a number of things to facilitate its entry. It's also initiating a signaling pathway that is loosening up the actin microfilaments. This one involves clustering of receptor protein tyrosine kinases, initiation of a signaling pathway that loosens up the actin layer again. There's another consequence of that activation of this receptor protein tyrosine kinase here, which is to increase the activity of this ATPase that pumps protons into the endosome. Remember, the endosome has to acidify, and that's done by pumping protons in. And so influenza virus binding stimulates the acidification. And then there's a third thing here. The virus apparently activates calcium channels, voltage-dependent calcium channels, which leads to increased import of calcium, which facilitates release of RNPs from the endosome. So 3 different things involved in entry there. And then on the right, Ebola virus binding to its receptor or to the cell surface, it leads to activation of PI3 kinase. And then there's the signaling pathway through AKT and RAC again. And the RAC activation actually inhibits the movement of the endosomes to these later endosomes, which I've labeled vesicle non-permissive for fusion. So it keeps the virus in the endosome where it can then fuse and the nucleocapsid can be released. So this is a beneficial signaling alteration, right? So that the virus can get out of the endosome, because if it goes farther, it can't get out of these vesicles. They're not permissive for fusion. And so by activating RAC, RAC inhibits this vesicle passage. The virus has a productive infection. So 3 examples of how signaling pathways can be manipulated to favor virus infection. This is all stuff we've learned recently. PI3 kinase is a very common focal point of many of these signaling pathways. And in particular, it's upstream of what's called mTOR, which is a complex of proteins that regulates protein synthesis or translation, as you can see on this slide. And so a number of viral proteins, 2 different slides here, hepatitis C virus proteins, adenovirus, rotavirus proteins, all bind to PI3K and activate it and send signals down to AKT. AKT in turn it allows the cells to live longer. It inhibits apoptosis, programmed cell death, which is good. The virus gets to multiply longer. Increases in metabolism, which you'll see today. We need energy to make virus particles. And through mTOR, increasing in translation. Okay, so that is one example. On the right is simply another with more viral proteins here. Hepatitis B virus, also herpes virus 8. G protein coupled receptor interacting with PI3K, and again inhibiting apoptosis, stimulating cells to divide, and stimulating translation. And now you see here, autophagy is a part of a response to stress where the cells wrap up pieces of the cytoplasm, and that generates membranes that are used by many virus reproduction cycles. So if you remember here, AKT is an inhibitor of apoptosis. This is going to figure in the next slide now. So this is a very interesting story where a viral RNA fragment promotes apoptosis, actually. So here we have a flavivirus that has a plus-stranded RNA genome, which is translated to give a very long polyprotein. These are envelope viruses with glycoproteins in them that are flat on the membrane. All right, so there's the viral RNA. At the 3' end, there's a whole series of stem loop structures. And this is a cartoon of the RNA here with the open reading frame, much reduced. It's not to scale. It's just to show you the 5' non-coding and the 3'. So in the cell, there is an exonuclease called XRN1. There are many exonucleases in cells, but this is one that we come to often in virus-infected cells. And this exonuclease chews from the 5' ends. It degrades RNAs. It degrades cellular RNAs because cell RNAs have to turn over, but it also degrades viral RNAs. It doesn't degrade all of the flavivirus RNAs, otherwise there'd be no infection. But it chews a fraction of them up, and it gets stuck at the 3' UTR. Because of all these structures, the XRN1 cannot chew anymore. And so what it leaves is a small RNA, sfRNA, small flavivirus RNA, it's called, 100, 200 bases long. And that RNA inactivates AKT. So remember, AKT is an inhibitor of apoptosis. So when you inactivate AKT, you now have apoptosis. You drive apoptosis, and this allows the virus to get out of certain cells. And in fact, if you take away this little sfRNA, if you make a little change in the 3' UTR, which prevents you from making this subgenomic RNA, so the XRN chews it all up, that virus doesn't form plaques. It will still grow, but it doesn't form plaques. It's the wild-type virus that makes sfRNA. If you take out the ability to make sfRNA, no plaques. So apoptosis is facilitating virus release from cells. So it's also needed for pathogenicity in mice. If you take this mutant that grows but doesn't cause plaque formation and you put it in mice, it doesn't kill the mice. So it's actually it's a candidate that's been used in some vaccines. It's a 30 base deletion, and it doesn't cause disease. And the virus still replicates, so it's a vaccine candidate. Very interesting. So seeing, again, looking at basic science, you can figure out solutions to making better vaccines. All right, so now moving on to gene expression. That's it for signaling. This is, of course, production of mRNAs, and we start with double-stranded DNA in the nucleus. We make a pre-mRNA, which is capped and polyadenylated. It's spliced and it's exported. And as all mRNAs have a fate, they are degraded at some point. mRNAs don't last forever. Our mRNAs have signals built into them that determine how long they last. They can be hours to days to weeks. Anyway, viruses interfere at each of these parts of these pathways. They can interfere with transcription. And in red are the viruses and the viral proteins that interfere. You don't need to know the virus proteins that do this. You just need to have this concept that viruses can interfere with transcription, with splicing, with polyadenylation, and with RNA export. That's what I want you to get from this. They interfere at every step. And then once the mRNA is in the cytoplasm, they can interfere with protein production, which we'll see in a moment. They can even mess with mRNA turnover. And why is this? Why would viruses have mechanisms to interfere with cellular mRNA production at multiple levels? Well, I think that this gives the viral mRNAs full access to the translational machinery. There's no competition with cellular mRNAs. So that's the reason for all these inhibitory steps. Reduce competition of cellular with viral RNAs for the translation machinery. So let's look at just one aspect of this in some detail. This is the nuclear pore complex here, where things are exported. Remember, we talked about how the nuclear export machinery recognizes splicing as a way of saying this mRNA needs to go out of the nucleus, because during splicing, a variety of cell proteins are deposited on the mRNA. They remain on the mRNA. That is, after splicing is done, all these proteins, our artist had a very fun time drawing all these. I like this one here, T-Rex. This protein is called T-Rex. And all of these are then bound by the export pathway proteins, and FX1 is one of them, and NXT, et cetera. So viruses, of course, they interfere with splicing, which I showed you on the previous slide, of cellular mRNAs. And that many viruses encode proteins that interfere with nuclear export. That's mainly what is shown here. For example, adenovirus has a variety of proteins, E1b and E4, that bind to export proteins. Poliovirus cleaves, processes some of these NUPs. Nuclear export proteins are needed to get out of the nucleus. It cleaves them so they don't work anymore. Poliovirus doesn't have anything to do with the nucleus, so it doesn't matter if its export is trashed, because its RNAs are mainly in the cytoplasm. It just is looking for unfettered access to the translational machinery. And influenza virus does this as well. So again, many viruses are interfering with gene expression. Viruses also interfere with mRNA turnover. Now, as I said, in the cells, there's a system for turning over mRNAs, a series of proteins to make sure that mRNAs don't last forever. And here is a schematic of what generally happens. We have an mRNA with an open reading frame. These mRNAs are capped at the 5' end. They have an open reading frame. They have a poly A tail. And there are exonucleases that can chew in from either end. So you see the one there at the top, CCR4, not PAN, is an exonuclease chewing in from the 3' end. And then to get it into the 5' end, you first have to take the cap off. The cap is protecting the 5' end of RNAs from this kind of exonucleolytic degradation. So there are decapping enzymes or decapping proteins, DCPs, which take the cap off, and then XRN, our old friend from the previous slides, can then chew from the 5' end. So both ends are chewed to get rid of these mRNAs. So this is a normal system in cells. And of course, these exonucleases are going to degrade viral RNAs, right? So many viruses encode proteins that inhibit their enzymes. So poliovirus, for example, degrades PAN, which is that 3' exo, DCP, decapping protein 1A, and XRN1. All three are degraded in poliovirus infected cells to prevent them from degrading the viral RNA. Okay, so this is one example. There are many examples of viral proteins that can antagonize these enzymes involved in mRNA turnover. Here's some examples, though, of going even further, where viruses take that degradation machinery and activate it so it degrades cellular mRNAs. And so here, for example, on the left is a cellular mRNA in the process of being translated. There's the 5' cap, M7GPPPNP. And the way translation works is that a protein in the cell binds the cap to get translation going. We'll see that in a moment. And these are all viral proteins that are basically enzymes that can cut off the cap. You see them here nipping at the RNA, and we have a SARS-CoV, the original SARS, human herpes virus 8 protein, herpes simplex virus. These are all taking off the cap, and once the cap is off, then XRN can come in and chew in the 5' end. And here's a vaccinia virus protein doing the same thing. It's a decapping protein taking off the cap so that XRN can chew it up. So these are examples of viral enzymes that make the mRNAs of the cell susceptible to degradation. Now, you may be thinking, why aren't the viral mRNAs degraded, right? You are thinking that, correct? Because that's a good question. What would distinguish the two? And it seems we don't have all the answers, but at least for some of these viruses, there are specific sequences in the viral mRNA that prevents their degradation by these enzymes. Otherwise, these processes would degrade both cellular and viral mRNAs. All right, the first question is, which of the following is a consequence of viral proteins modifying signal transduction pathways to promote viral replication? Poliovirus inhibition of transcription by RNA polymerase II, herpes simplex virus protein blocking pre-mRNA splicing, disrupting actin filaments to allow endocytosis, initiation of mRNA degradation by viral proteins. What's an example of viral proteins modifying signal transduction pathways? Disruption of actin filaments is the right answer. So that's a signaling pathway. Everything else is not a signaling pathway. The inhibition of transcription, blocking splicing, mRNA degradation, those are all just enzymes acting on RNA or processes, but they're not signaling. The disruption of actin filaments is like on these two slides. Disruption of actin filaments is a consequence of a signaling pathway. The inhibition of transcription, the inhibition of splicing, the degradation of RNAs, those are not signaling pathways. The signaling pathway is we have a series of events. A virus binds a receptor, and then a series of phosphorylation events, a signaling cascade inside the cell occurs. Now I want to talk a bit about protein synthesis. This is a major regulatory point in virus-infected cells, both by viral proteins and by the cell. And so remember, the Baltimore scheme puts mRNA at the middle because all viral genomes need to produce mRNA that can be translated by the host cell, because the host cell translation machinery comes from the host only. It's not viral. Parts of it may be viral, but the whole thing is not viral. So viruses need to make that magic mRNA. So all of our 7 classes go to mRNA. And the way mRNA is translated, we need to know this before we proceed further. This is a very quick primer on how mRNAs are translated. We have an mRNA at the top. There's a cap at the 5' end, and there's an AUG codon that specifies initiation of protein synthesis. A big complex of proteins assembles at the 5' end, and it's nucleated. The assembly is stimulated by the binding of this protein, EIF4E, to the cap structure. That is crucial because then all these other proteins bind in a row. 4G binds to 4E, and then EIF3 binds to 4G, and then the ribosome, the 40S subunit, binds to EIF3. These are all protein synthesis initiation proteins. So the cap is essential for recruiting the ribosome to the mRNA. And then the complex starts to move down from the 5' end in a process called scanning. And when it reaches the AUG codon, then what's called the ternary complex, which is basically a met tRNA with a number of other proteins associated with it, and the anticodon UAC, which matches the AUG. That comes in and binds, and then the 60S subunit is recruited. So now we have a full ribosome on the mRNA with the very first tRNA there, the met tRNA, and then the ribosome can start moving down and decoding the mRNA one triplet at a time. That's how translation is initiated. All right, so this is important to remember. Many viruses turn off protein synthesis in infected cells. Obvious reason, again, to reduce competition for the translational apparatus. So here is a graph showing you the rate of protein synthesis on the y-axis at times post-infection. These are cells infected with poliovirus. And what you see is by 2 hours post-infection, the rate of protein synthesis has declined substantially in these cells. And then at about 3 hours, it starts to go up again. And eventually the cells die at 5 hours or so, and there's no protein synthesis. So what's happening here is there's a shutoff of protein synthesis by the virus. And how that happens, I'll show you in a moment. And then at 3 hours, that's viral proteins being made. Shut off the host cell, you make viral proteins. So now you've got the whole translation apparatus to making viral proteins. That's the modus operandus of this, I think. And on the right is a protein gel. So on a protein gel, you can separate proteins by molecular weight. And what we have here is 1, 2, 3, 4, 5 lanes where the proteins have been taken at different hours after polio infection. So 0 hours, 1, 3, 5, and 7. And you can see at 0 and 1 hours is just a smear, right? Because there's so many cell proteins there. They all migrate right next to each other from the biggest to the smallest all the way at the bottom here. So they're all cell proteins. But you see then at 3 hours, you start to see the smear going away. The smear is a very technical, accurate biological term, smear, right? Like a bagel with a schmear, right? It's the same idea. You get a smear of proteins on a gel, because you just have so many that you can't distinguish them. But now the smear is going away, and now you're starting to see individual viral proteins which you can identify. And at 5 hours, there's much less of cell protein synthesis. It's all replaced by viral proteins, and then the cells die, so they're not making many proteins at all. So that's an example of shut-off or viral inhibition of cell translation or cell protein synthesis. So I use protein synthesis and translation equivalently. And so how is protein synthesis turned off? In that example I showed you, here's the mechanism, actually multiple mechanisms. Now remember, at the bottom, to make protein, you have to have an mRNA with a 5' cap so that the EIF4E can bind and then bring in all these other proteins that eventually bring in the 40S subunit, all right? That's why you need a cap. So in cells infected with poliovirus and foot and mouth disease virus, which are related viruses, they have a protease encoded in the genome that cuts this red protein, EIF4G, so that most of it falls away from the 4E. So 4G binds EIF4E. That's how 4E recruits 4G to the mRNA. So now these viruses have a protease that cut the complex, so you can see that if 4E binds, there's not going to be any 4G and there's not going to be any ribosome. So that's what shuts off protein synthesis, the cutting of EIF4G. So that's one mechanism. There are others. Adenovirus and influenza virus dephosphorylate EIF4E. It turns out that you need a phosphate on 4E for it to bind tightly to 4G. And if you take the phosphate off, the 4E falls off 4G. So it's kind of functionally equivalent to cutting 4G, but it's just a different mechanism. So dephosphorylation. And finally, a third mechanism, there is in cells protein called 4EBP1. Cells need to regulate translation, and this is how they do it. 4EBP1 will bind EIF4E. And in that state, 4E cannot bind 4G, so there's no protein synthesis. So if the cell needs to turn off protein synthesis, this is how it does it. The binding of 4EBP1, and that stands for 4E binding protein number 1, is regulated by phosphorylation. When there is a phosphate on 4EBP1, it cannot bind 4E. And so some viruses dephosphorylate 4EBP1. That allows BP1 to bind 4E, and that shuts off host protein synthesis. And now, the virus can have access to the translational apparatus. So now you're surely asking yourselves, how are viral proteins made when 4G is cleaved, or when 4E is dephosphorylated, or when 4EBP1 is dephosphorylated, right? You are asking yourselves that, because it's a good question. Why isn't this shutting off viral protein synthesis? Well, I'm going to tell you now. Viruses have structures at their 5' ends that bypass the need for many of these initiation proteins. So, for example, cellular mRNAs need all of these proteins. They need 4E and 4G and EIF3 and the 4DS subunit, of course. And they have to bind to the cap. So cellular mRNAs require all the eukaryotic initiation factors, or EIFs as they're called. Many viruses do not need any of those. In fact, poliovirus and related viruses, I've already told you that they cut the 4G, so they don't even need a cap. They don't need EIF4E. They do need the 4DS subunit, and they do need EIF3, and they do need 4G, but only this part of it. They don't need EIF4E. So these viruses have found a way to translate in the absence of 4E, and the mRNAs don't need a cap. And I'm going to show you how that works in a minute. It's actually shown here. This 5' end of these viruses is highly structured, and that structure can bind 4G directly. You don't need a 5' cap to recruit 4E. So that's one answer, how you can get a viral message translated when 4G has been cleaved by viral proteases. And then at the bottom, hepatitis C virus also has, these structures are called irises, by the way, internal ribosome entry sites. And hepatitis C virus has one that will bind EIF3 directly without even 4G or 4E. There's no cap on it, binds EIF3 directly, and then the 4DS can bind it. So really a minimal requirement for initiation proteins. And so that's how these viruses can inactivate 5' end recognition and still have their own mRNAs translated. All right, so this process of having ribosomes not need the 5' cap is called internal initiation. And was first discovered in the genome of poliovirus. So many years ago, 1981, before some of you were born, I think. Yeah, definitely before many of you were born. I sequenced the genome of poliovirus, and at the top, I had to type it out by hand. We didn't have computers back then. And that was the sequence I got. And there's the AUG codon, and there was all this other stuff in front of it. There's the 5' end, base 1. And we didn't know what to make of all of this. We had no idea. How did the ribosome find this AUG? And it turns out that this is an internal ribosome entry site. It recruits ribosomes in the manner I've just shown you. So let me show you how that works. So these areas of these genomes, these 5' UTRs, are highly structured. So here's the poliovirus 5' uncoding region. I didn't know it had this structure when I had the sequence. It's just a sequence of letters. You know, you have to figure out if it's structured or not, and there are ways you can do that, and I didn't do it. But that's what it looks like. It has all these stem loops. And then we subsequently found that other viruses have similar 5' non-coding regions. They're given names type 2, 3, 4, 5, 6 iris because they have different structures. But what they all do is allow the ribosome to bind internally. Forget about the 5' N. You don't need a cap. You don't need EIF4E. The ribosome can bind internally. And so, for example, on that second one, the type 2 iris, the 4G binds this little region right here of the stem loop, and then the EIF3, and then the ribosome can come in. All right, so that's how viruses can turn off cellular translation and still make their own proteins by what I've called an iris. There are other ways as well, but that's a very striking way to do it. Okay, so viruses turn off cell protein synthesis, and their own synthesis obviously continues, and that's one of the mechanisms by which that happens. Cells, in turn, also try to turn off viral protein synthesis. I want to tell you now how that works. As a defense mechanism, cells and viruses have evolved together for many, many years, and so both have tried to outdo the other. You know, we call this an arms race. A virus tries to do something, and the cell overcomes it, and then the virus overcomes it. It goes back and forth over many, many, many years. So what does the cell do? And that's embodied in this slide here. So this is the initiation step of viral protein synthesis. We have eliminated all of the binding to the cap, and what we have here is now the methionine tRNA bound to the initiating AUG. There's the 40S subunit. And when the 60S ribosome comes in and binds, what is released from this is EIF2 with the molecule of GDP, okay? So EIF2 is an initiation protein that comes in with the tRNA. So there's the tRNA with the methionine. And you see EIF2 there has GTP, is guanosine triphosphate bound to it. But when the 60S comes in, that GTP is hydrolyzed, the energy is needed for the subunits to come together. And now you have EIF2 with GDP. But you need more EIF2 with GTP, so you have to make it. You have to recycle this GDP because there's limiting quantities of EIF2 in a cell. And so whenever you take off a phosphate, you have to put one back on. So the way that's done is by this protein called EIF2B, where what happens is EIF2 binds to it, and then the GDP is exchanged for a molecule of GTP, and now we have GTP EIF2 that can go back with a tRNA and do another round of translation. Right? So this is basically, this EIF2B is what we call a guanine exchange factor. It holds on to GDP and then exchanges it for GTP. Very important because it allows you to have enough GTP EIF2 for translation. So what does the cell do? In the cell, there are protein kinases called EIF2 alpha kinases. And there are three of them shown here. There's one in the ER, there's one that responds to amino acid deprivation, and then there's one that responds to double-stranded RNA. So it basically conditions that mimic a virus infection. So endoplasmic reticulum stress, when viruses start to make a lot of glycoproteins in the ER, that's a stress condition that cells can sense, and that kinase called PERK is activated when cells are full of double-stranded RNA, which happens when viruses replicate. It activates a EIF2 alpha kinase called PKR. I'll tell you how it activates them in a moment. Basically, what those kinases do is they phosphorylate the EIF2 alpha subunit of EIF2. So remember, this is now in response to a virus-infected cell. EIF2 alpha gets phosphorylated. And now, when GDP EIF2 phosphate binds EIF2B, it can't be recycled. It can't have its GDP swapped out for GTP because that phosphate locks it into EIF2B. Right, so the phosphorylation of EIF2 alpha, a subunit of EIF2, is a response to turn off protein synthesis. And this is global. It's everything, viral and cellular. And you could imagine that the cell is going to sacrifice itself so that the virus infection doesn't spread, because obviously turning off your protein synthesis isn't a good thing for the cell. But that's the mechanism. The EIF2 phosphorylation inhibits the initiation of protein synthesis, which is universal among viruses and cells. So how does this work? Cells, let's look at PKR. Cells have a gene that encodes PKR. It is translated as an inactive protein. Here it is inactive PKR. It has a kinase domain, which is the part of the protein that puts the phosphate onto EIF2 alpha. And then it has two double-stranded RNA binding motifs. These are parts of the protein that bind double-stranded RNA. Why double-stranded RNA? Most virus-infected cells contain double-stranded RNA. Whether it is an RNA virus-infected cell, it's made by reproduction of the genome, you get both strands. Even in DNA virus-infected cells, you get double-stranded RNA, because if you have a double-stranded DNA virus, both strands are being made into mRNA, and those are opposite polarity, so they can hybridize. So you get double-stranded RNA. That's the signature of virus infection. The double-stranded RNA activates the kinase. As the kinase is made, it's called PKR, protein kinase RNA activated. As it's first made, it is inactive. It is unable to phosphorylate anything. These molecules bind double-stranded RNA via their double-stranded RNA binding motifs. They then phosphorylate each other. And then that makes them active so that they can go off and phosphorylate EIF2 alpha. So the idea is you have double-stranded RNA, multiple PKR molecules are binding double-stranded RNA. They are phosphorylating each other to activate each other, and then they go off to phosphorylate EIF2 alpha. So that's how the activation works by double-stranded RNA. So to summarize, PKR is actually induced by virus infection. It is one of the interferon-induced genes that we'll talk about in a couple of weeks. It is activated by virus infection. As soon as there's double-stranded RNA made, PKR is activated. It inhibits host translation by phosphorylating EIF2 alpha, and it also induces programmed cell death. The phosphorylation of EIF2 alpha is a signal for induction of programmed cell death, so the cell's killed, and hopefully that stops virus infection. So this is a very powerful mechanism, right? Yeah, it is. Every virus around has an antagonist of this. Otherwise, they don't exist. It's so powerful. Every virus that we know of has some kind of antagonist of this PKR pathway. So let me give you a sense of that. Here's one encoded in the adenovirus genome. It's called VARNA1. And its structure there is shown on the left in brown. And what it does, it binds this RNA. It's a short RNA. It binds a single molecule of PKR. One molecule only is bound by VARNA. What do you think happens? You need two PKRs to phosphorylate each other, bound to one double-stranded RNA, as we have seen and here shown on the right. But VARNA is short. Only one PKR can bind at high affinity. No activation of PKR, no phosphorylation of EIF2, protein synthesis continues. So this is an RNA made pretty early in adenovirus-infected cells, and as the virus is making double-stranded RNA, before it can activate PKR, the VARNA is made, and that prevents its activation. So protein synthesis continues. It has to, otherwise the virus is finished. So that's one example. It's a small RNA that binds PKR, prevents its activation. And here's just a list. You don't have to memorize this list. You just have to know, you have to look at it and go, wow, look at all those mechanisms for countering inactivation of EIF2 by phosphorylation. So here are the different players. We have EIF2, which is the target. We have one of the kinases, the ER kinase called PERK, which senses ER stress, and we're going to actually come back to this today. When this protein is normally present as monomers in the ER membrane, and when ER stress occurs, it dimerizes, phosphorylates itself, and then it becomes a kinase that can phosphorylate EIF2 alpha. And then here's PKR itself. So we have all these different antagonists at different steps. For example, vaccinia virus encodes a protein that looks like EIF2, but it's not. So PKRs all go to it and phosphorylate it, but they ignore the real EIF2. Variety of viruses antagonize PERK directly, the kinase. There are double-stranded RNA binding proteins that will bind the kinase and prevent it from activating. VARNA1 is one of those, and there are antagonists of the proteins directly. So you see multiple viruses encode different mechanisms for antagonizing this because it's very powerful. And if it's not antagonized, the virus simply doesn't exist. Okay, so that is another example of how the cell tries to turn off translation globally, which would stop infection, and the virus counters it. And there are other mechanisms as well, but that's a very cool one that I like. The other thing the cell does is to form what are called stress granules. And in a cell, such stress granules can form when a cell wants to turn off translation. The cell shunts all of its mRNAs into this stress granule where they're no longer active. And when they're needed, they can be pulled out. And so a stress granule is shown there in yellow. It has mRNAs in them. Some of them are in the process of being translated. And then there are structural proteins, G3BP, TIA1, TIAR. These are all structural proteins that make up the stress granule because you can see these in either an uninfected or an infected cell. Now, of course, if viral RNAs are put in a stress granule, they're not going to be translated. So yes, viruses encode antagonists of stress granule formation. And again, they're listed on this slide. You don't need to know the specific antagonists. But I just want to show you, for example, what's a good one? Here, this is pretty cool. Hepatitis C virus, in order to reproduce, assembles lipid droplets in cells, and we're going to look at this in a moment. And among the proteins that are part of this lipid droplet complex, there are viral proteins, the RNA polymerase, G3BP. So the virus steals D3BP, so it can't go to the formation of stress granules, so that's how it inhibits stress granule formation. Now, poliovirus cleaves G3BP. It has protease called 3CPro that cuts G3BP so it can't form a stress granule. And other viruses have interesting ways of antagonizing them as well. So the point is, when a cell senses it's infected, it's a stress response is to form a stress granule, and the virus can then prevent them from forming in order to enable its genomes to be, its mRNAs to be translated. So at every step, when the cell does something, the virus counters. When the virus does something, the cells counter. So there's this constant to and fro, if you will, between virus and cell. And you can imagine, as we heard before in the question, these genes vary in human populations. They're not all perfect. So some of you can have suboptimal forms, and maybe you're more prone to infection. Maybe you get more severe infections. It's all possibly related to these countermeasures. PKR is an interferon-induced enzyme that is activated by blank, leading to phosphorylation of blank and inhibition of translation. So the blanks can be GDP, EIF2 alpha, double-stranded RNA, EIF2 alpha, double-stranded RNA, EIF2B, single-stranded RNA, EIF2 alpha, none of the above. Double-stranded RNA, right, and the PKR is activated by double-stranded RNA, leading to phosphorylation of EIF2 alpha. A lot of you picked C, double-stranded RNA, but it is not EIF2B that's phosphorylated, it's EIF2 alpha. EIF2B is the protein that will bind irreversibly the phosphorylated EIF2 alpha, and then GDP cannot be recycled. Let's talk about metabolism. Remember this? Remember the Krebs cycle, the TCA cycle? I learned this in high school, and I thought I'd never see it again. And now I'm seeing it again because it's important for virus production. It's amazing how much energy you need to make virus particles. Of course, you need to make nucleotides, amino acids, fatty acids. It takes 4 ATP to make a single peptide bond. Amazing, and you're making thousands and thousands of virus particles per cell. So basically, viruses have to crank up metabolism of cells in order to reproduce. So let's explore how that happens. First, there's increased glycolysis, the breakdown of glucose. Very simple experiment. We have cells here, either mock-infected, infected with adenovirus, wild type, or a mutant that doesn't reproduce. And we're looking at different times after infection. And this medium has a pH indicator in it. It's red at neutral pH, and as the pH drops, it turns yellow. And you can see wild-type adenovirus, the pH seems to be dropping around 36 hours, and then it gets lower and lower, it gets yellow. That's because what's being made is lactic acid, the end product of glycolysis. And on the right is simply a graph showing the actual consumption rate of glucose in these cells, which you can measure. You don't have to just look at the cells. You can get a number by measuring the consumption of glucose. And you can see that adenovirus consumes glucose quite readily, whereas the mutant, which doesn't reproduce, the medium's not turning yellow, and the glucose is not being consumed. This is typical for many viruses. There's a paper called Rhinoviruses Have a Sweet Tooth. Rhinoviruses, common cold viruses, also require glucose for reproduction. So what happens here? There's the glycolytic pathway. You start with glucose at the top. It's imported into cells by transporters. That's the GLUT134. And then it undergoes a series of metabolic steps, ending up at lactic acid. And there are a couple of shunts off of this pathway that we can talk about. But of course, glucose is the major breakdown of dietary carbohydrate. It is eventually converted to pyruvate here, giving 2 ATPs and 2 NADHs. So that's not very much ATP, but some of these intermediates allow more synthesis, as you can see here. And this pathway is perturbed in virus-infected cells, as I'll show you in a minute. But one of the aspects I want to point out here is on the way from glucose all the way down to pyruvate and then eventually lactic acid. There was a shunt here to the right called the pentose phosphate pathway. So glucose 6-phosphate can be removed from this glycolytic pathway to give rise to ribose 5-phosphate, and eventually is a precursor for synthesis of nucleotides. So this pathway can also supply precursors to nucleotides, which of course are needed to make nucleic acids. And here's an example of what viruses can do to the glycolytic pathway. These are 2 different herpes viruses, human cytomegalovirus, herpes simplex virus 1, both of which infect all of us. At this point in our lives, we all have these 2 viruses in us. And they have 2 different effects on glycolysis. So cytomegalovirus stimulates the uptake of glucose and brings it through the glycolytic pathway down to acetyl-CoA, which is a precursor for fatty acid synthesis. So fatty acids are made from acetyl-CoA. Herpes virus, on the other hand, doesn't stimulate the uptake. It deals with normal amounts of glucose uptake. It does not bring it to pyruvate, but rather shunts it off that ribose 5-phosphate pathway that I noted just previously. So herpes simplex appears to prefer the synthesis of nucleotides rather than lipids. And one of the ideas for this is that it has a shorter reproduction cycle, and so it's more important for it to synthesize DNAs relatively quickly, and it can use the normal amounts of lipids that are found in cells to make its membrane, whereas cytomegalovirus, much longer reproduction cycle, needs to stimulate membrane biosynthesis. Perhaps CMV can deal with the amounts of triphosphates that are found normally in cells. Now, there's an interesting aspect of the glycolytic pathway. Acetyl-CoA, that end product, is produced in the mitochondria from pyruvate. but it cannot cross the membrane. It can't get back out. And so it has to get back out to participate in fatty acid synthesis pathways. And so it is converted to citrate in the mitochondria. So acetyl-CoA is converted to citrate. The citrate can be transported out by a transport protein, and that can be then converted back to acetyl-CoA and then eventually to other precursors of fatty acids. And so these are important for viruses that have membranes. In fact, not just membranes in their particles, but depend on membranes on which to reproduce the nucleic acids. And so most, for example, human cytomegalovirus infected cells, most of the citrate that comes out of the mitochondria eventually goes towards making fatty acids. If you inhibit this enzyme, the malonyl-CoA synthetase, you inhibit the production of human cytomegalovirus proteins. And vesicular stomatitis virus, which also depends on membranes, also tinkers with another member of this shuttle. So this is an essential part of the increased biosynthesis for many viruses. These changes in glucose metabolism can have consequences for human disease. And so these are two viruses that infect the liver. We call them hepatotropic viruses. We have hepatitis B virus there on the left and hepatitis C virus on the right. As you know, the liver is the site of glycogen synthesis and also what we call gluconeogenesis, the de novo synthesis of glucose. Infection with hepatitis B virus is associated with type 2 diabetes, the type which involves insulin deficiency or resistance. And that's because virus infection induces the enzymes that are involved in glucose synthesis. So the virus requires glucose for production of energy, as I just showed you, but this has a pathogenic consequence because it leads to type 2 diabetes. Same occurs with hepatitis C virus, but a different mechanism. It reduces glucose uptake from the cells, but also increases gluconeogenesis. So these alterations I'm telling you about are not just of academic interest for understanding how the viruses reproduce, they have consequences for disease. That brings us to the citric acid or the TCA cycle. I'll never forget how my biology teacher described this in like high school? Yeah, it was high school. She wouldn't mention ATP. She just said puffs of energy, she called it. And I never understood what a puff of energy was. What is this cloud that she would draw on the board? It was ATP. Why didn't she just say ATP? It would have made much more sense. As you know, the citric acid cycle takes in pyruvate, product of the glycolytic pathway that I just showed you, converts it to acetyl-CoA, and then it goes around the cycle, converting it to different steps, and different amounts of energy are produced around the cycle. So this is an incredible hub of carbon metabolism. It gives you precursors for many, many different things, fatty acids, and pyrimidines, and purines. It makes energies, and viruses impact the cycle. For example, just showing you here, these are a couple of herpes viruses and vaccinia virus. So cytomegalovirus pulls off citrate here to make fatty acids. So it would normally halt the cycle, but it pulls in glutamine to alpha-ketoglutarate to make up for the loss of citrate. And herpes simplex does the same thing. It pulls out the oxaloacetate to aspartate to make pyrimidines and purines for its nucleic acids, and it increases the uptake of glutamine to compensate. Otherwise, the cycle would halt if the virus is simply pulled off these intermediates without replenishing them. Okay, so that's a very quick example of how the TCA cycle can be altered by virus infection. Let's talk about lipids now. Lipid metabolism. Now, in most of us, lipids are the triacylglycerols are the primary energy source. We oxidize them to get energy, and we also store them in case we need them. We store triacylglycerol. So there on part A is a triacylglycerol. It's the building block for lipids. These are in the circulation bound to lipoproteins, or bound to proteins, and they're called lipoproteins. And to be stored in the cell, They are degraded by lipoprotein lipases on the plasma membrane to form free fatty acids. Each of the chains, of course, is a fatty acid. Fatty acids are imported into the cell. They're converted to acyl-CoA, and then eventually to triacylglycerols, which are incorporated into lipid droplets. And those are the storage depots of lipids in cells, these lipid droplets. And then when they're needed, the lipids can be removed and oxidized for energy. And viruses, we're learning, are manipulating these pathways to provide energy. So let's take a look at a couple of examples. This is cytomegalovirus. Cytomegalovirus, to build the virus particle, apparently requires very long chain fatty acids, very different from the normal composition of our membranes. And so infection increases the uptake of glucose, as I showed you previously, which leads to acetyl-CoA, and that eventually will lead to the production of lipids. And so the way it works is quite interesting. So the genes for fatty acid and sterol synthesis are, of course, in the nucleus, and they're controlled by promoters that are regulated by this transcription protein called SREBP1M. That protein has to bind this promoter in order to turn on the genes that are needed to make lipids and fatty acids. And so in a cell, normally they're turned off because this transcriptional activator, SREBP1, is stuck in the endoplasmic reticulum. So there it is, SREBP1, and it's a form called P. It's stuck because it's bound to a couple of proteins, and this one is the one that's keeping it in the ER, INSIG1. So how does it get out to get in the nucleus and turn on fatty acid biosynthesis? Remember, when viruses make glycoproteins in the ER, it causes ER stress, and that can be sensed by PERK, that protein in the ER that dimerizes when there's ER stress. So just the synthesis of more than the usual amount of protein in the ER causes this stress. It activates PERK. PERK dimerizes. It then pulls INSIG off of this complex, including SREBP1, and it can now start to move towards the nucleus and eventually get in the nucleus. So basically, the virus is making a lot of glycoproteins. It's causing ER stress. It's activating PERK, and then PERK pulls off the inhibitor of SREBP1, which is INSIG1, and then SREBP1 moves through the Golgi and eventually into the nucleus. It turns on the genes for fatty acid biosynthesis that are required for building virus particles, and particularly long-chain fatty acids for assembly. And other viruses do similar things. This is another example. I want to show you the balance of synthesis and oxidation. This is hepatitis C virus infected cells. So on the bottom left, we have cells on the left, mock-infected cells, and on the right, virus-infected cells. And they're human hepatocytes, so that they are the target cell for hepatitis C virus. And they are stained with a dye that stains lipids. It stains them green. And you can see there's an increase in the green staining comparing the infected to the mock. So virus infection somehow increases the accumulation of lipids in cells. So how does it do that? Lipid metabolism is a balance of synthesis and oxidation. So synthesis, of course, is lipogenesis, and oxidation is the degradation or the use of the lipid to make fatty acids. And so what the virus infection does is to increase lipogenesis and decrease oxidation. And the net effect is an increase in lipids in the infected cells. Now, there are two separate pathways. One on the left is involved in lipogenesis, and the one on the right is involved in oxidation. And essentially, there are genes in the nucleus that are driven by a promoter that turns up that SREBP protein, which in turn will drive lipogenesis. And the virus, so this particular transcription factor, FOXO1, is needed to drive the synthesis of proteins that lead to increased amounts of lipid synthesis. Normally, this transcription factor leaves the nucleus in most cells and it's not active, virus infection inhibits its exit from the nucleus. So now there's more FOXO in the nucleus, and you have more lipogenesis. So that accounts for the increase. And if you knock down the FOXO1 mRNA with siRNA, for example, you inhibit replication, as you would predict, because increased lipid synthesis is important for virus multiplication. On the other hand, the oxidation of lipids is carried out by proteins that are also encoded in the genome and driven by a promoter, in this case, which is responsive to a transcription factor called FOXA2. And Hep C infection lowers the amount of FOXA2 in cells. And if you overproduce that protein now in cells that are infected with Hep C, you drive oxidation and you inhibit Hep C replication. So you can see the virus is increasing synthesis, decreasing oxidation, and the net result is that you make these lipid droplets which are needed for virus multiplication. Last question. How might virus infection lead to increased levels of ATP? Stimulation of glucose uptake, increased glycolysis, increased oxidation of fatty acids, increased utilization of glutamine, or all of the above. All right, let's see how we did. All of the above is the answer. That's right. All of these things can lead to increased levels. Stimulation of glucose uptake, which generates ATP in the pathway, the glycolytic pathway, increased glycolysis, increased oxidation. Oxidation of fatty acids provides energy in the form of ATP, and increased utilization of glutamine. Last couple of slides, I want to tell you about how, this is about cellular organelle remodeling. Viruses remodel organelles in order to provide replication platforms for the nucleic acids. And so here in poliovirus-infected cells, we see a similar experiment. We see increased import of fatty acids into virus-infected cells. These are virus-infected cells stained with a dye that turns green when fatty acids are present. Mock-infected cells, much less import of fatty acids. And so what happens is in a virus-infected cell, in a polio-infected cell, the membranes are converted into these double membrane replication complexes on the surface of which the RNA is synthesized. And we talked a bit about that when we talked about RNA synthesis. And the raw material for these membranes comes from other membranes in the cell. The virus stimulates lipolysis, breakdown of the lipid droplets into fatty acids, which are then made into phospholipids and used to build the replication compartment. And a series of viral proteins shown here, 3A and 2C, bring the lipid droplets to the site of where these replication compartments are going to be built. So they anchor the lipid droplets. Remember, the lipid droplets are the storage form for lipids in cells. The virus basically is taking out the components and putting them to build these double membrane replication compartments. And this is a common theme in many viruses. Here's hepatitis C virus, where we saw stimulates lipogenesis, inhibits oxidation of lipids. These viruses, again, assemble on membranes. And early in infection, you see the formation of these membranous structures that are derived from the endoplasmic reticulum. You see these structures here, which are left open at one side so that viral proteins can come in. All these blue and red and yellow structures are viral proteins. The viral RNA is produced in the lumen of these structures. And eventually, lipid droplets are built in these structures, and those serve as the platform for assembly of new virus particles. You can see a particle there is budding into the lumen of this membrane, and it's encapsulating a piece of RNA with it. And eventually that virus particle will make its way to the cell surface. So again, lipid membranes essential for reproduction, in this case assembly. And you can see in a cell, if you take electron micrographs, you can see an extensive network of these vesicles budding off of the endoplasmic reticulum. This also happens with coronaviruses. Coronaviruses are making both mRNAs and genomes on double membrane vesicles. Here's an electron micrograph of those double membrane vesicles. These, again, are induced by viral proteins. All of these viruses that are inducing double membrane vesicles, they're made by the action of individual viral proteins. And again, it's on the surface of these vesicles that RNA synthesis occurs. Again, this is done to concentrate all the components in one place so that they're not all diffuse and around the cytoplasm. They're on the surface of vesicles. It makes the reactions more efficient. And finally, one more example where I think illustrates the amazing effects of evolution. This is a plant virus called tomato bushy stunt virus, because those are the symptoms it induces in tomato, makes them short. And this virus reproduces its RNA in, again, vesicles that are produced from the ER, very much like the virus we just looked at, except these are closed off vesicles. And RNA synthesis occurs here. Now, this virus brings in pyruvate kinase into the vesicle to make ATP that's needed to make nucleic acid. It supplies the energy for viral RNA synthesis. And in particular, one of the viral proteins called p33 coats the interior of this vesicle. p33 binds pyruvate kinase specifically. It imports it into the vesicle, and there it can generate the energy that is needed for RNA synthesis. PK supplies ATP for the helicases that are needed to unwind the RNA. And these are some nice immunofluorescence micrographs where we're looking at p33, which is in blue fluorescent protein. You can't see it very well, but there it is there. And then we have green fluorescent protein labeling the pyruvate kinase. It's in green. And here you see one of these membrane vesicles coming off the ER. And the merged, you can see both the blue and the green color are together, showing you that the p33 and the pyruvate kinase are together. So membranes are very important for reproduction of many viruses. All right, so next time we're going to go from infected cells to infected organisms and understand how viruses cause disease and how we can prevent it.